acetyl group, you often turn to acetyl-CoA. But when it comes to moving a methyl group, methionine provides a way. And this way is through the molecule S-adenosyl methionine, or adoMet, sometimes called SAM-E, and it's one of the key ways in biochemistry in which we could transfer these one-carbon groups, these methyl groups. And this allows us to methylate things in the process of making various hormones like melatonin and adrenaline, as well as to methylate proteins. So we can methylate the histone proteins that our DNA is wrapped around. This allows us to alter the accessibility of regions of our DNA in order to make it so that we only make certain proteins at certain times. In both the case of our acetyl-CoA or any sort of CoA linkage, or the case of our S-adenosyl methionine, the reason why we're able to move these sorts of functional groups or these various chemical pieces off of these kind of carrier molecules is due to the sulfur linkage. So sulfur is right under oxygen on the periodic table. This means that they have the same number of valence electrons, so the same number of their most reactive electrons, those in the outermost shell. But in the case of sulfur, they're going to be farther out. And so those are going to be more reactive, more nucleophilic. And it's also going to be that because sulfur is so big and bulky, it's harder for it to form effective bonds. Remember that making a covalent bond involves orbital overlap. When you have a sulfur, it's basically kind of like someone with a really big hand. And then if you try to have them shake a little kid's hand, say, that's going to be kind of a weak shake. The same sort of thing. When sulfur is bound to one of these smaller elements, the bonds that it forms are going to be weaker, and therefore they're going to be easier to break. So we're going to be able to swap off groups, such as swap this methyl group off of our S-adenosyl methionine and onto something else, which allows us to do all sorts of cool biochemistry. In order to keep using methionine this way, however, we need to keep regenerating the methionine after we transfer the methyl group, and this is going to involve some folate biochemistry. We can also use methionine in order to make cysteine. So lots of cool stuff about methionine. Let's dig into the details of this methyl moving. How we're going to do it is that we're going to use a substitution mechanism. So we're dealing with nucleophiles and electrophiles. And so if you need to review those, go back and review them. But here's just a quick summary. A nucleophile is something that has more electrons or electron density than it can comfortably handle. And it's going to seek out an electrophile, which is something with which it can share that electron density with. We call it a nucleophile because, well, if you think about what you have in a nucleophile, you have more electrons or electron density, so you have a partial or a full charge, negative charge. Negative charges will seek out positive charges. Where do you find positive charges? Well, you find them in the nucleus, where the protons are. So a nucleophile will attack the nucleus of an electrophile. Well, it'll just attack the electrophile. And what it can do then is it can form a new covalent bond. It can have things like substitution, elimination, or deprotonation. What we're going to be dealing with is a substitution reaction where we have a nucleophile attack an electrophile and kick off a leaving group. You formed a new bond to one part and you've kicked off a leaving group. In order for this to actually happen, well, our nucleophile has to actually want to attack and our leaving group has to be kind of happy on its own. In the case of moving a methyl group, if we were to just move it off of methionine, with this acting, we were going to have like a nucleophile attack this carbon. Well, we would need this carbon to be a stronger electrophile. And if we want this leftovers to be happy on its own, well, we wouldn't want to just kick off a methyl group because then what we'd be left with would be like a thiolate, which would be then reactive. And so that's not what we want. Instead, we want to activate this methionine to make this part a better leaving group and to make this part a better electrophile. The way that we're going to do this is we're actually going to spend energy. We're going to be making it using this methionine adenylyl transferase we'll talk about. We'll make it so that we make this sulfur positively charged. When we do this, what's going to happen is now this sulfur being positively charged is going to start draining electrons from the things that are next to it, from this carbon that's next to it. Sulfur is already more electronegative than carbon. So what this means is that in those bonds where the carbon and the sulfur are forming those bonds, they're going to be weaker bonds like we talked about because the sulfur is bigger. And because sulfur is more electronegative than carbon, because it's farther to the right on the periodic table, it's not as much, it's more electronegative as oxygen because oxygens grow up, but sulfur is still more electronegative than carbon. And so it's going to be able to kind of win the tug of war battle for those electrons, especially because the sulfur is extra desperate because it has that positive charge. What's going to happen now is that this carbon being partly having this partial positive charge is now going to be vulnerable to attack from a nucleophile. And then that nucleophile attacks, you can get this transfer. So and also this SAH, so your kind of leftovers once you've kicked off the methyl group, well, this is going to be a better leaving group than if you just had a methionine without this adenosine. Basically, you would have a thiolate, you would have a negatively charged sulfur, which would be vulnerable, which would then be attacky, and so it wouldn't be a good leaving group. But this is a much better leaving group. We have this neutral molecule that's less reactive. So how do we actually do this activation step? This is happening with the help of methionine adenylyl transferase. So this is actually a pretty wacky, cool mechanism. The methionine, the sulfur is actually going to attack the innermost part, the part where the phosphate groups are actually attached to the adenosine. Normally, when we see ATP being used, we see just like one of the phosphate groups being used. And so we can kind of just like burn ATP and kick off a phosphate group to get ADP, or we can actually use the attach part of the ATP on. So we might attach like an ADP part on or an AMP part on. So you might see a molecule attacking one of these oxygens. In this case, however, what we're attacking is going to it's actually going to be the carbon on this adenosine. So we're going to get this S-adenosyl methionine where we've attached this adenosine part on, but through this sulfur. So it's this pretty wacky linkage, really cool. Don't have time to get into the mechanism or anything like that here, but I'll post a link where you can go look into it. But again, this is kind of like this really weird reaction because we're attacking the adenosine part and we're actually kicking off all of 3 of these phosphate groups. And we're kicking them off though. We're going to hydrolyze them to give us this PPI, so this inorganic phosphate, and then this orthophosphate, this PI, and then this inorganic phosphate can further hydrolyze, and then this can give you basically a big energy boost. So you're paying a lot of energy, but you're really activating this S-adenosyl methionine molecule. Once we have this activated, well, now we can move it somewhere. In order to move it somewhere, basically, we said we had this carbon is now going to be more electrophilic, and so we need a nucleophile to attack it. Some places that we'll find nucleophiles are going to be things like hydroxylate groups or thiolate groups, amine groups. Those are going to then have a partial or full negative charge. They're going to have a lone pair of electrons. They're going to be able to form a bond to this carbon. When they form a bond to this carbon, basically the bonds that sulfur was sharing with that carbon, now sulfur gets to keep those electrons and it's going to now be neutral. Being neutral, it's going to be less likely to kind of go backwards. And so remember that the thiolate would act as a nucleophile itself, and so that would not be a good, it would not be good to be left over with something that was then going to act as a nucleophile and attack the carbon. So we don't want that to happen, but in the case of our SAH, well, now we have this form that's going to be neutral and happier as a leaving group. When we get these reactions, they're typically happening through an SN2 mechanism. We have our nucleophile attack and form a new bond while the old one breaks. There are also some methyltransferases that use radical mechanisms, and I'm not going to go into them here, but I will post a link to a paper if you want to learn more. Now we have our methylated thing. That's all great, but we're also left with this molecule SAH, S-adenosyl homocysteine. In order to activate this to actually use it again, we're actually going to split it up, remake methionine, and then remake this SAM. So we'll use this adenosyl homocysteine in order to cut off the homocysteine from the adenosine. We can then use the adenosine to make more ATP and do various things. And then we could take that homocysteine, methylate it to get us back to methionine, and then we can activate this and do it all again. So we have the adenosyl homocysteine that's kind of cutting things apart. And then we get our methionine synthase, which is going to give us the methyl back to reform methionine. To actually get that CH3 group, we're not going to take it from SAM. Instead, we're going to take it from one of our other common one-carbon sources, which is going to be folate. So folate, there are various forms of folate, and they can contain various carbon groups that can be transferred. And you'll actually see that serine can form a source of one-carbon groups as well, which can modify the folate to kind of activate the folate to activate other things. So we see lots of interplay between various amino acids, between various forms of folate, all sorts of cool stuff. In this case, we're using this 5-methyl tetrahydrofolate in order to regenerate our methionine from our homocysteine. And remember, the homocysteine itself was coming from hydrolyzing the S-adenosyl homocysteine to give us our homocysteine and our adenosine. Now we've taken the homocysteine and we've added the methyl group back on, and now we have this S-adenosyl methionine again. So we can do this whole thing again. In terms of actually moving that methylating the methionine, well, that's what we saw in the very beginning. An alternative fate for that homocysteine that we make is to generate cysteine. So we're still going to take that SAH and we're going to hydrolyze it by using adenosyl homocysteine in order to give us our adenosine and our homocysteine. But then what we're going to do is we're going to use cysteine beta synthase in order to combine homocysteine with serine in order then cysteine lyase in order to get us cysteine. So if we look at what's happening here, we basically take this homocysteine, and if you imagine flipping it over so that this is the part that comes from the homocysteine, and this is the part that came from the serine after we kicked off the water. If we want to generate a cysteine, we need this top part of the molecule. So we need to kick off this bottom part of the molecule. So remember that methionine, basically we had 2 carbons, a sulfur and a carbon. We kicked off a carbon. So in our homocysteine, we don't have that, but we still have this like 1 too many carbons. Rather than try to take a carbon out of here, we're just going to put the sulfur group onto the serine. We're going to do this by combining the two and then cutting them apart. So we flip over this homocysteine in the diagram. We've attached this part of the serine. Now we want to kick off this part as cysteine, and this part will be our leaving group. We do this using cysteine lyase. This is going to leave us with this weird molecule called alpha-ketobutyrate, not to be confused with beta-ketobutyrate that we see in our ketone bodies, as well as this molecule of cysteine and that amino group, that nitrogen group that we kicked off. So this will give us cysteine from our methionine. So cysteine and methionine are 2 amino acids that contain sulfur, but they're very different in terms of their reactivity and their properties. Cysteine has that thiol group. It can form that thiolate that we were trying to avoid forming with our methionine so that we had a better leaving group that wasn't going to attack In the case of cysteine, we can make it a good nucleophile. We can make it a tachy by deprotonating it to give us our thiolates that can serve as a powerful nucleophile. In the case of our methionine, well, here instead of our thiol group, we have a thioether group. We have the sulfur with carbons on either side. And as we saw, we can activate it using ATP to give us S-adenosyl methionine. This is going to make it so that this methyl group is going to be a stronger electrophile and that this SAH group is going to be a better leaving group. This makes it so that we can then transfer this carbon group, this methyl group, onto various things, which allows us to, say, synthesize our hormones, as well as to modify epigenetically. So lots of cool things that are coming from methylation, but you're not going to be methylating from a cysteine. You will be methylating from a methionine. But cysteine can still be used to do really cool stuff. It can form crosslinks as well. It can sop up some oxidative stress. So lots of cool stuff with cysteine, but it's very different from methionine. But methionine can be used to make cysteine. Cysteine cannot be used to make methionine, however. In fact, methionine is going to be one of our quote-unquote essential amino acids, meaning we have to eat it from our diet. Yes, we can regenerate it through from this S-adenosyl homocysteine, so we could regenerate it from like homocysteine, but on net, we can't make it. We can't make new methionine, so we've got to get it through our diet, hence calling it an essential amino acid. But remember, all of the amino acids really are essential because we really do need them. But it's just like dietarily methionine is dietarily essential as well.